this biosensor or chemical sensor, okay, is similar, right? The term analyte, the term receptors, be it bioreceptors or chemical receptors, and the specificity of the receptors, okay, which is very important in development of sensors. If it is not specific, if it is not selective, then it's useless in terms of accuracy. Okay, so the study of bioreceptor or chemical receptor, or some, after this I'm going to use the word biomarker or chemical marker, is the same. The study of that biomarker and chemical marker itself requires a long time. It might be 5 years just to get the right bioreceptors or biomarkers or chemical markers. Okay, it might be if it is a new disease, if it is a new analyte of interest that you want to study, then it requires a long study to achieve that. Okay, so we also discuss a bit on types of transducers, but this will be in detail after the biomarker. I think it will be next week. We will learn about electrochemical and also after that is optical. Okay, so this part has to be understood first, understood well, before we can proceed to the rest of the biosensor and chemical sensor part. The general structure is like this. It has to be. Okay, so I've shown you examples of sensors. Okay, the problems, some of the problems comes during the development of sensors, and these are our main targets. Okay, this is what we want to achieve when we wish to develop the biosensors or chemical sensors. We must make sure that it is compliance to this objective. Okay, and then I also explain some important parts apart from the fundamental part just now, is on the sampling and the fabrication. Of course, the biomarker is also important. It's very important indeed. All right, and then I mentioned about the AI assisted sensors. Okay, so we can integrate AI. Okay, there are many softwares that can help us to increase the accuracy in development of biosensors. I also have given examples where the sensors can be. Okay, it can be not just, it cannot, it's not limited to medical, but it can be in environment, it can be in water analysis, it can be in agriculture as well. Okay. Right, food packaging, okay, food quality. I mentioned about samples and also sampling because those are very important in terms of practicality. How do you sample? What kind of samples suits the sensors? Okay, if it is very difficult to acquire the samples and requires a very difficult sample pre-treatment, then it's not going to be practical. It has to be, the word is for medical is non-invasive, means that you don't have to draw blood, trigger pain to those who wants to do the analysis. Okay, might as well we use something easily obtained, like sputum, sweats, urine, saliva, tears. And I have introduced to you the usage of ISF, which is quite new, interstitial fluid, which actually third point in equilibrium with capillary blood, meaning that it can represent the actual, not all probably, biomarkers that exist in the blood. Okay, because blood is well studied relatively to urine, sweat, and other bodily fluid. Yeah. And I also have introduced to you wearable sensors. Well, you cannot find it yet in the market, but it is currently happening. And in future, yeah, soon we are going to have it, probably. Okay. All right. And this is the part that we have discussed a bit last week on DNA. Three different types of biomarkers. You can see that recognition molecules, okay, there are many terms, bioreceptors, biomarkers, recognition molecules. The meanings are the same, okay, the terms are quite similar, okay, and then these three are among commonly used. Okay, there are others, of course, there are others like enzymes, like cells, like short protein, okay, and will in the end shows about chemical markers as well. But these are quite common. And I have shown you the DNA, okay, the basic DNA structure with phosphate backbone, okay, the base pair, which is very important, the one that determine the selectivity, which is the base pair. Okay, and the bases in the DNA are ATCG. This is the amino acid. Yeah? Amino acid ATCG. But what about RNA? What's the difference in terms of the bases? Anyone can help me? Which one?
Strand.
Yes, help me. Yes, help me. Are you trying
Single strand.
the strand, single
Single
strand?
strand, single strand, and the RNA didn't have thymine.
Yes, it is. What is
Uh,
what's
you uracil.
yes,
Uracil.
yes, very good. Okay, so RNA is very important. So instead of DNA, we can also use RNA for development of biosensor, but unfortunately, RNA is not stable. Okay, so that's the biggest issue when we use RNA, if we are to use RNA in development of biosensor, because it's not stable. RNA has to be converted into cDNA first, which is not favorable if we are to develop sensor, which we want it to be user-friendly. We do not want the hassle of processing before we can do the measurement. Okay, so that's why RNA is not really favorable in development of DNA based on, biosensor based on DNA. But there are biomarker based on microRNA, miRNA. Okay, so this is a different principle. Okay, probably towards the end of these courses, we will study recent trend using microRNA, using CRISPR, which difference then, which is a bit different on how the detection scheme is done. Okay, because microRNA also is a good biomarker, but it's not going to be similar to what we will discuss today. All right, so hydrogen, this is just to discuss, to show to you how the interaction between the bases, it is based on hydrogen bond, okay, between the amino acid between the bases, okay, right? So I also have shown examples of perfect match, okay, and possibility of mismatches, yeah, possibility of mismatches because of the sequence. So remember that it has to be A with T and C with G. So if it is in the strand, it has different sequence, then there is possibility of uh, mismatches, and you have not perfect match. Okay, probably you have 50% match, 75% match, or 30% match. Okay, and that determines how strong the hybridization. It's very important to understand the hybridization concept because this is the one that give the selectivity if we are to develop DNA-based biosensor. This is the principle of how it works. Okay, the reading or the conclusion from the sensors, if it is based on DNA, will rely on the hybridization. Okay, so let's say if we have 100% match, then of course we are sure that the analyte of target that we are trying to measure is there in the sample. Okay, let's say we have 50% match. Means the sequence is not 100%, it only match 50%. So how do we conclude? Okay, so this is a very big issue. Probably if I can say, if you want example, E. coli. So E. coli have different variants as well. Okay, forgive me if I use wrong words. I'm not a molecular, I'm not a bacteriologist. Okay, so they have different strands, different variants, just like, okay, dengue, for example. Okay, so dengue have 4 serotypes. Okay, means that dengue virus have 4 different variants. Okay, so each of these variants has specific sequence of DNA. Specifically for dengue is RNA. Yeah, they has these 4 variants, these 4 serotypes have different sequence of RNA. Okay, they match certain sequence, but they do have a different sequence. Okay? So for dengue, it's not important if you have like serotype 1 or serotype 2, because the most important one at this moment is whether you are dengue positive or you are not with dengue, with something else. Okay, so it's not really important to know whether you are serotype 1 or 2 or 3 or 4. Okay, unless the symptom, probably not the symptom, the seriousness of the symptom will be based on serotype, then probably that will be an issue. Okay, so how do we get this 100%? How do we get this 100%? So this is something very important in development of DNA biosensor. Okay, the word DNA probe. Okay, why is it important? Because I don't know any of you has done PCR analysis. Okay, probably during that pandemic time. Okay, we are used to rapid test kit, but some of you might have experience doing PCR. Okay, because it is more accurate compared to rapid test kit. Okay, so PCR requires, the term is primer. Okay. So what is a primer? It is not the same as probe. For development of DNA-based biosensor, the probe, the required one is DNA probe. It's not primer. Probably the development, the preparation of DNA probe and primer, probably they have some similarities during the process, but it's not similar. Okay, what is a probe? A probe is a single-stranded sequence of DNA, or of course RNA is single strand, used to search for its complementary sequence in a sample genome. Okay, the probe is placed in the contact. We say, okay, this is a process. So how do we know the sequence is unique? Okay, so in every species, they have certain regions, certain regions of DNA that defines us, that defines the species. Okay, so in order to get the right sequence of DNA probe, I will show you what does it look like. This is the example of DNA probe. Yeah. So this DNA probe is specific for dengue, I think. I don't remember from where I took this one. So this DNA probe is specific for dengue virus. Okay. So how do we get this? It has to be designed. There is a software. Okay? There is a software that can be used to gain all this sequence. Okay. Particularly the one that we want to have is this one. Okay, up to here, right? So this is the one that beyond my capability to explain how the designing is done, but it is by using software, and that software also has to search for the sample genome, meaning that species, we need to have the whole sequence of that genome of that particular species. For example, just now I mentioned about E. coli. Okay, so in order to design a DNA probe for E. coli, the whole genome sequence of E. coli has been established. So the software can do analysis and generate a sequence. For example, this one is for dengue virus, that can represent the unique gene sequence of dengue. Okay, that is why it is called sample genome. That is unique to that particular species. Okay, can you get that? Right. Okay, the word is complementary. Okay, so the DNA probe that we design, okay, before we can develop further into DNA biosensor, has to be complementary sequence in terms of ATCG just now, with the sample genome region. So there is a region. That one you have to talk to molecular biologists to know the process of producing or designing the probe, and to understand the specific region that define the species. Okay. So this is very important. Okay. So it's not just any DNA, any sequence can be used. No, we have to make sure that we get the right DNA probe first. Okay. So when we did this dengue biosensor, since there are a lot of established research on this DNA probe, so this is based on established review, journal papers that has been reported before. Okay, so there are of course some different. If you use different probe, you will get different results. Okay, because these probes might be different, designed by different people, even though using the same software, but probably the design, the way they design it different. Okay, so we have to test first whether there is a match, or there is no match, or there is 50% match and whatnot. Okay, so before we develop the biosensor, we have to do it synthetically first, because we don't directly test with the actual virus. So you can see here, we have two types of probe. Okay. One, you can see the sequence are all similar, GGGG, okay, and probably the endpoint. This is not one of the sequence. This is just what we call as a linker. The C6H12 SH is referring to the word thiol. Okay, same goes to SH. So we need a linker. Why? I will explain to you why we need a linker, because in order for us to put the DNA on top of the sensor, we need to have chemically linked to the sensor. And you probably observe there are many other types of linker. This is thiol. This is aminated. Okay, depends on what kind of material that you are working with. We will learn this after the biomarker. We will learn about methods of immobilization. So that will be another section. Yeah. All right. So as I mentioned, we do not test directly with dengue virus first. During the development stage, we use synthetic complementary DNA. So if you check the probe, okay, with a complementary, you will see exact match. Okay, so if you look at here, it's AG. It does not, it goes sideways. It goes here. Okay, AT. It's opposite side, because if you can imagine how the DNA right. Okay, this is the 5' end. Okay, it will start to react with the 3' end. Yeah, so I don't know how to do it. It goes here. Okay, so that's why if you look at the probe, the matching starts from this end. Probably this is 3' end. This is 5' end. It moves opposite side. Okay, and we also have to synthetically simulate, yeah, the mismatches. You can see that there is one mismatch here. Okay, why we do this, we want to make sure that the reading, when we get the reading later, how do we see the signal if it is 100%? How do we see the signal if it is 50%? How do we see the signal if it is just one mismatch? Or how do we see the signal if it is 100% no match? Because we need to do some signal calibration. Okay, so we need all this synthetically first before we can test on actual virus. So far, so good on DNA probe. Any questions? Okay, all right. So this is an example of how we attach. Okay, this is basically, we are going to revisit this during transducer part. So you don't have to think about, okay, how does it work? But enough for you to look at example, how we start to do DNA-based biosensor. But this one is based on conductometric. Okay, but you do not have to worry about the words. Okay, so we start with the sensor platform, regardless what kind of sensor, and then you need to put the probe. Let me use different color. Where's the color? Okay. In this case, this is the DNA probe. Yeah, this one. Okay. Okay. Now, so you can see now the linker. That is the usage of the linker because we need to put it standing up. If the DNA lies down, then the process of hybridization is very difficult. Okay. Now, in real world, when we test this biosensor for real virus, for example, or any other food samples or plant samples, okay? Same goes with blood samples or sputum samples or saliva samples. We have to do extraction, unfortunately. For DNA biosensor, we have to extract the DNA out from the sample. Okay, so that is one of the hurdles, the difficulty when we have to do biosensors because that has to be done outside. Okay, but you can do it integrated, but we don't want to discuss that yet. So let's say you have saliva samples. Let me change the color. Okay, let's, we want to use saliva and we are testing the virus in the saliva. Okay, but we already have this DNA probe on the surface of the sensor. Okay, so chemical process of extracting DNA from saliva, not the saliva itself, DNA from the virus exists in the saliva. Okay, so let's have it in red color. Okay, now the next process is to mix them together. Means that I'm going to mix the extracted DNA into the sensor. Okay, so depends on what kind of sensors, but this biochemical process requires buffer, requires a bit of heating. Those are something that we have to comply. Okay, so once they start mixing, these are the things that we need to observe. Whether you have 100% match, whether you have 50% match, or no match at all. Okay, so this is what happened. You can see that the green one is the DNA probe, and the red one is extracted DNA from the virus. Okay, so if it is 100% match, the extracted DNA with the DNA probe embedded on the sensor, then of course you will have changes, okay, changes of signal. Okay, so that's why when I mentioned we need to design synthetically first, what kind of changes if we have 100% match? What kind of changes if we have 50% or 10%? Okay, because we need to know these changes that we observe is due to 100% or is due to 50% or it might be due to 10% of matching. Okay, so we have to conclude from that signal. So if it is 100% from that signal, giving you an indication of 100% interaction, 100% match, then of course you are sure that, okay, this is the right virus that we are targeting. Okay, this is positively that virus that exists in the saliva, for example. But if it is a 50%, okay, we know that it might be the same virus, but different variant might be. Okay. So if it is 10%, then probably it's not that virus. Probably it is something else. Okay, right? So those indication, okay, those indication are very important. Those conclusion are very important. based on this signal. Whether, yeah, it's positive, whether, or it's probably a different variant, or it's no match at all. Yeah, it's different thing that exists in that saliva. Okay, so that is the fundamental of how we develop DNA biosensor. I'm not talking about what kind of transducer yet. Okay, we are talking about the basic principle. If we are to use DNA or RNA, if it is possible, biosensors. Okay. So any question before we continue discussion? Yes, quite heavy. Okay. This is just to give to you a bit detail. Yeah. So I hope that you understand now. This is a bit detail on the construction, the fabrication. Okay. So this is the DNA probe. Okay. In this case, we are using electrochemical, and this is electrode. I hope that everybody at least remember the word electrode from your previous knowledge. Okay. So you can see the process of putting in, attaching the DNA probe first with all the chemistry. Okay. So this is when I come in. Okay. Remember that developing this kind of sensor requires molecular biologists to design the biomarker, and then the chemist in the middle, and the third one, those who do electronic, the engineering part, and also the probably AI part, yeah, to assist the reading. Right, so DNA probe, just now, you have to make sure that you understand, which is the most important part because that determines the selectivity. Okay. So this is the extracted DNA from the samples. Okay, any sample, saliva, blood, plants, food, or any others. Okay, and then the hybridization process. So once that is happening, then we will detect whether the signal generated from this electrochemical device is giving us 100% match, 50% match, or no match at all. Yeah, before we can make conclusion. Okay, everybody? Now, I hope that you probably understand 10% of DNA biosensor. Yeah. So I don't want to jump into the word intercalator, this linker, not yet. Okay. But up to here down, you can see the process of hybridization and the importance of DNA biomarker. And in order to develop DNA biosensor, we need to have DNA probe that is selective, that is representing the genomic sequence of that particular species. Okay, before the next process. Okay, so this is just me. I just want to show that apart from electrochemical, it can be optical. You can see the similarity in terms of you have to put the DNA probe first, and then next the hybridization happen. It's just that the signal generation is based on fluorescence signal or probably color generation. So this is the one that differentiates between the transducers. Transducers is the method that gives out the signal. Yeah. All right. So I don't want to talk about this because this is a bit advanced. All right. Are we okay on that? We are now at 6:36 before I introduce another biomarker. Any question on DNA? So you probably are familiar when you do like watch criminal movies that they want to know about the DNA, yeah, to confirm the identity of probably the perpetrator, the murderer, or to confirm the identity of the deceased, the one being murdered, yeah. The same principle actually, but they are using PCR, which is benchtop device, and they are using what we call as general primers. Okay, so the same similarity check, yeah, whether the matching is 100%, 99%. If it is 99%, it's confirmed. Okay, so those is the same principle. Yeah, it's the same principle. Okay, if there is no question, I just proceed with the second part of biomarker. In here, we are talking about the word immuno. Just now is DNA. This one is immuno. And the biomarker used is antibody. Okay. So anyone familiar with antibody? Actually, I can't remember all your background. Okay. As I mentioned, probably last lecture, the production of antibody is quite difficult. Okay, the DNA just now is chemically produced. It's actually chemically produced. Once we know the sequence, the DNA, the bases, then we can synthesize because it's all chemical. The amino acids are all chemicals. We can just synthesize. But for antibody, it has to be induced. Okay, at this moment, uh, uh, we have to use in vivo, yeah, process, but they also meaning that the one that I knew, the antigen has to be injected into animals, okay, the animals that use rabbits, mice, okay, and the antibody produced from those animals are harvested, okay, harvested. So it's a quite, I would say, sad because the animals usage. But now there are certain ways of doing producing antibody using recombinant process. I know some of the principle, but I don't want to discuss that because I might be wrong because I'm not, again, a molecular biologist. So by doing recombinant production of antibody, it avoids the usage of animals, yeah, which is good because the problem that we researchers face when we work with antibody harvested from the animals, the performance of the antibody differs from batches. So I don't know what's the factor. So now if you look at the structure of antibody, again, it's all about chemistry. This is the carboxyl group. Okay, it's Y-shaped. Okay, so carboxyl at the this, I don't know what to say, the bottom one. Okay, and you have amine, okay, on top. And the one that is important to control is and do the binding is here. Okay, the amine site. Okay, the carboxyl will not do any binding. Okay, only the amine site. Yeah, the here. Okay, that binds with the analyte. Okay, so this is how antibody works. I think from during from I think from schools or from your undergraduates, you will probably have heard of antigen antibody interaction. They are very specific, like lock and key. Okay. Okay, so this is where the antigen will interact. Okay, what is antigen? We know already antibody, the Y-shaped molecules. Okay, so look at here, this is bacteria. Okay, so antigen can be a lot of things. It can be, okay, the easiest that we can, I can give examples, the easiest is bacteria, virus, or any proteins can be antigen. Okay, so let's look at bacteria here. Bacteria. This is the cell wall. The purple one is the cell wall of the bacteria. Okay, and you see there are different shapes of. You have a cone, squares, a cube. Sorry, you probably observe spheres. Okay, so this antibody, because it's very specific, it will only interact with certain epitopes. The word here is epitopes. Okay, so the different shapes that you see represent different epitopes on the cell wall of the bacteria. So this is well known virus that attacks us starting 2019, end of 2019, and we start to have pandemic globally in 2020, starting March. So this is the face of that particular virus. So you can see that this is what we call antigen. This virus is antigen. But which part of that antigen interact with the antibody is this, the S protein. So this is the, probably the epitope. Okay. There are other protein as well that can interact with other types of antibody and protein. So if you are to design a test kit, a diagnostic kit, the antibody for S protein will be different. The antibody for N protein is different because it's very specific. Okay, right? So this is the face of an antigen. All right, and this is also antigen. Okay, and they have specific epitopes. Okay, that react with specific antibody. So that's why if you look at the issue of SARS-CoV-2, we are very familiar. Every time we have new variant, new variant, new variant. Okay, why? Because the S protein is not stable. Yeah, because they are RNA-based virus. So RNA is not stable. The S proteins easy to mutate. Okay, and that gives us different variants. The N protein is more stable. But you can see the location of N is a bit difficult to access. So the detection most of the time is based on S protein. And since it is not stable, it's difficult actually to detect because the sequence are mutating. Okay, I hope that I have presented the understanding of antigen-antibody interaction and how specific they are towards certain epitopes, yeah, certain epitopes where the antigen reacted with the antibody. Okay, and this is types of antibodies. Okay, yeah, this is a Y shape, and this is the antigen. So polyclonal antibody is more general. It will react with probably generally more epitopes. Okay, but for monoclonal antibody, it is more specific. Okay, there are certain applications that requires monoclonal. There are certain applications that polyclonal is much more preferred. That depends on the design. Okay, what you want to do. Of course, the production of monoclonal is much more expensive compared to polyclonal. Yeah. All right. Okay. All right. So probably we have gone through this. Okay. Yeah. The pandemic taught us a lot. When you submit your samples, you can choose whether you want to do PCR test, which is more expensive, or you can do the rapid test kit based on antigen. Okay, so these two will give you results. Of course, the PCR test is more accurate, but requires probably 1 day if you have the right instrument. But antigen test you can have like in less than 15 minutes, 20 minutes. Okay. So, but in terms of accuracy, this might have an issue, the antigen test. Okay. The antibody test is not really practical because this is useful for past infection because the antibody in our body will be developed after some stage. Okay. So the usage of antibody test is more on to check the prevalence, to study the prevalence, means that how serious the infection is in terms of these cases, number of cases. So because some has the infection but does not show any symptom, but based on the antibody analysis of the blood, probably determine that he or she already infected but show no symptom. Okay, so the antibody test at this moment, this is just for past infection. So you can see it is used for population screening. It's not for a check on whether you are positive or negative at current infection. Okay. So how does it work? One last one. How does it work? Yeah, it's quite easy for this one. It's not based on hybridization or whatnot. Okay, it's based on interaction. So the correct antibody, if it finds the exact antigen, it will react. It will interact. It will not react with any other antigen which is not designed for that particular antibody. So the principle is the same. The principle is easy. It's not based on hybridization, but is based on interaction of the antibody, the Y shape, with the antigen specifically for that antibody. Okay, so how the signal is generated to give us the whether the antigen and the antibody has interacted correctly. That one, there are a few. Okay, colored base, color base, electrochemical base. We will learn about it in transducer. Okay, but enough for me to show you. Of course, the easiest one is the rapid test kit. Okay, the rapid test kit is based on the usage of gold nanoparticles. The color that you observe, the line that you observe is actually the color of gold nanoparticles. Okay, so you can see that when it match, okay, the virus, the antigen is there, then you have the test line positive. generated by the color. So if there is no color, you only have the control line, there's still color produced on the control line only. Okay. So this is just examples of work that we have done on tuberculosis based on antibody. Okay. So if you are interested, you can go through this during your recess from your 7 to 8 time. Okay. All right. So any question before we take a break? No question. Okay, everybody, let's take a break and I'll see you at 8. Bye.
Okay, Prof.
The recording. Right. So the last, as I mentioned, the 3 common biomarkers that I will discuss in detail, a bit detail, what will be DNA, antibody, and aptamer. But I will introduce to you probably today the other types, but only superficial. Will not be able to discuss detail. So I hope that you have understood the difference between how DNA biosensor work and antibody work. They use different principle. DNA based on hybridization, whether it has 100%, less than 100%, or no matching at all. Okay, so that's the basis of how we detect the analytes. Analyte in this case, it's easier to be as an example, virus, bacteria, or any other pathogenic, any other species. For antibody, it is based on interaction between antibody and antigen. So antigen is defined again similar. It can be virus, it can be bacteria, it can also be protein. Yeah, not necessarily the whole bacteria, the whole virus, but it can be a small protein. Right. So what is aptamer? So aptamer is a single-stranded oligonucleotides. So oligonucleotides, so if DNA consists of amino acid and bases, the ATCG, so same goes with aptamer. Okay, it consists of a series of amino acid called oligonucleotides. When we have a series of amino acids, it is called oligonucleotides. Yeah? Amino acids is nucleotides. When we have a series of nucleotides, it is oligonucleotides. Okay, but the keywords here is folded. Yeah, the behavior of folding gives the selectivity of aptamer. So if you look at here, example of the folding. Yeah, right. So what's so special about aptamer compared to DNA and compared to antibody? Okay. So easier if we can discuss relative to compared to antibody, because antibody requires the process just now, the in vivo process. It has to be induced. The production of antibody has to be induced at this certain technology. You can do recombinant, but most of the time it is induced in animals. So what if you have other types of toxicant, for example, drugs? pesticides, or you have metabolites. Okay, which will not trigger antibody, production. Okay, so let's compare aptamer with DNA, for example. So if you look at the same example, there is no way that we can get a DNA probe for drugs, for pesticides, for metabolites, for example. Okay, even though these are toxicants, for example, drugs, pesticides, they are very important to be detected. So it's a toxicant that requires us to analyze. So if it is DNA, it requires another DNA. The target must be DNA. If it is antibody, the target must be antigen. So this one does not fit in the requirement for using DNA or using antibody. So the option is designing an aptamer. Okay, so just imagine that this is sort of if we are in chemistry, we are used to the word ligand. And one of the example of ligand that I discussed last week is EDTA. Okay, so EDTA, ethylene, diamine, tetraacetic acid can react with metal ions. Okay, so forming chelates. Yeah, so I hope that you remember your chemistry. So that's how ligand or chelating agent works. It forms probably electrostatic interaction between the lone pair electron in the oxygen, in the nitrogen, with the metal ions. So they form a complex. Okay, that is a ligand or chelating agent. But aptamer probably works the same way, but now we are talking about a series of amino acid. And we have to understand that this is also a chemical compound. When we talk about amino acid, you probably observe they have carboxyl, they have amine, definitely is a mass. Okay, and then we know that they contain lone pair electron. And some amino acid has thiol group, yeah, sulfur, thiol, we call it thiol group. So these contain in amino acid, different kind of amino acid has different functional groups, chemical functional groups. So we can design this aptamer to be a selective marker. We define it biomarker because it contains amino acid for this kind of analytes. Okay, so this is a good option. Okay, if you don't have anything that can have DNA, or you don't have anything that can trigger the production of a selective antibody, then we can use aptamer. Okay, so I hope that uh, I hope that you have understood the difference between DNA, antibody, and aptamer. Okay, so how do we get the selectivity based on aptamer? Is through the folding process. Okay, so protein, they love to fold. Okay, so how does it look? Okay, so if we look at this example, okay, if we look at this example, okay, this is antibody just now. Okay, so specifically it will react with antigen of interest. But if we have aptamer, okay, you can see there is a specific folding structure, and it can interact with also protein, antigen if we design it well. Okay, it is by design. It is by design. And the good thing is, it is based on chemical, it can be chemically produced. You don't have to use animals to produce aptamer. You can, but it's easier if we can produce it in the lab, okay, synthetically. So aptamer can be produced synthetically in the lab. Okay, so this is the process. Yeah, it is called selects process. I don't have it, the full name, full abbreviation. This selects is an abbreviation. Eh? This is not on the slide. I will try to show you another slide to what is selects mean. So this is a process, okay? So there is an in vivo selects, and we don't want to discuss it, but this is something that you can do in the lab, okay? Via chemical reaction, okay? So this is not something that I want to discuss in detail, but the process is doable. It's something that you can do in the lab, and you have to check the selectivity. Through this process, we can have a lot of aptamers sequence, but we have to test it. Which one gives the best binding, which one will give the best selectivity. Okay. This is an example of structure of aptamer. You can see the folding. Yeah, you can see how they interact. So this is the analyte in the middle. Okay. This is the analyte and the oligonucleotide, which is the aptamer, folded surrounding the analytes of interest that we want to detect. So that gives the interaction, the specific interaction. Specificity of aptamer will be determined on how good we design it. Okay, so nowadays we can do this via wet chemistry. Okay, this is select systematic evolution of ligands by exponential enrichment. So that is full name of selects. Okay, we can do this via wet chemistry, means that you mix something in the lab, but we can also do this using in silico. In silico, meaning that you can design the aptamer not by using wet chemistry, but using computational approach. Okay, you can, that one is beyond my knowledge, but it's doable. You can design the aptamer just by using computational. Okay, so there is a software to do that, and there is a process for you to study in designing the aptamer. So this is something very good, very versatile. It gives a lot of option. Okay, if you have a new analyte that does not have DNA, does not have antibody, does not have any enzyme specifically that can react with the analyte, then we can try to design aptamer. Okay, so same process. Okay, if we have any kind of sensors, we have to put the aptamer on top of the sensors, and they will interact with the target, with the analyte. So how do we detect the interaction? That's up to the transducer, whether we are using electrochemical, whether we are using optical, color, or fluorescent, or UV absorbance, depends on the interaction. That one is another discussion. Okay. Yeah, so this is different types of how aptamer is being applied in development of sensors. Yeah. So I'm not going to discuss detail here, but enough for you to understand. For example, here we have different viruses on the same strips, but in using aptamers, because if you design it well, it is very selective. You can see that it can detect a series of virus different with different aptamers. Okay, so at the same time, you can detect many diseases using a single strip. Okay, so that's why this is very important uh, to explore aptamer, because it has many application. It's not limited to only those with DNA, and it's not limited to those that can produce antibody. So it's more robust. Yeah. Right. Okay. Before we discuss a few other examples of biomarker, I would like to come back to ionophore. Okay. So this one is considered chemical marker. Okay. Chemical marker. So what is ionophore? So you can see this is one of example of ionophore and how it looks like. The structure is a bit complicated. It is a compound that form complexes with specific ions. Yeah? So chemical marker, usually we work with ions. So this one is for potassium, if I'm not mistaken. And how to prepare this? You need to have a very good knowledge in synthetic chemistry. Okay, you have to do the synthesis. You have to prepare the ligand. You have to mix it. You have to until you get the structure. Yeah, so it's actually very challenging. Yeah, to produce ionophores. It's very challenging. And this is another example. The name of the ionophore called lunactin. And this ionophore is very selective towards ammonium ions. So you can see the structure is complicated. Yeah, it's complicated. It's like a loop structure that can envelop the ammonium ion. This one also looks complicated. All right. Yeah. This one also looks complicated. So that's how the selectivity comes in. Yeah? It has to be complicated. If not, then it will be very general. Okay, so this is how it works. It can. This is not something that you have to worry. It can help to transport the ions through the membrane. Okay, so if you look at the structure, you observe a lot of oxygen, carboxyl ketone or carboxyl with lone pair electrons. So that's how they interact, through electrostatic interaction of ions, positive ion, and negativity of lone pair electrons. So that's how they interact. So those are ionophore. Okay. Right. Let me. Okay. Stop share this one. I will share on a bit on extra on aptamer. Okay. Touch this. So before we talk about CRISPR, talk about Aptamus a little bit more. So, okay. So this is the application of aptamer using lateral flow. So we can use aptamer to produce lateral flow. And this is an example of metabolite. Okay. Yeah. Sorry. So this is a project by my PhD students. She's working in chemistry department. The problem is for her to identify cyanide poisoning. The issue with cyanide is it is not stable. I mean, once in the body, it will metabolize. It will metabolize into metabolite. Secondary metabolite. So probably this is primary metabolite. I'm not sure. So it's difficult for her to analyze, right? The probably the sample from I don't know whether she investigate murder, but the possibility of cyanide poisoning, because the cyanide will no longer be in that body to be analyzed. But what we can analyze is more on the metabolite after the cyanide degrade or metabolize into a metabolite. So the problem is here, how do we design the biomarker? Because it is a metabolite, we do not have any specific DNA, we do not have antibody, and at the same time, we also do not have a chemical reagent that can react to the metabolite. So the metabolite is ATCA. The full name is 2-amino 2-thiazine 4-carboxylic acid. So it's a metabolite. So that's why aptamer in this case comes into picture, which is more practical, right? Comes into picture because we need to design the biomarker, right? So she uses the SELEX process. Yeah, very difficult. It was a very tough project. But she is a very diligent student, so she managed to get a series of aptamers. Yeah, I think, yeah, a group of aptamers from the SELEX process. And in order to identify which aptamers has the best binding, has the best selectivity, of course, she has to do some analysis. Yeah, so you can see that the production, the binding activities has to be studied in order to choose which aptamer. Okay, so this is like the series of aptamer that she produces from the SELECT process, and she runs some analysis, identified 2 aptamers that is highly selective towards the metabolite. To confirm on that also, she did some computational. Okay, so this is how the aptamer looks like. It has a folded structure. You can see that most of it will look like this. Okay, so she also did computational, studying the binding sites. Yeah? So this is the analytes, okay, the metabolite, ATCA. And she studied the binding sites via computational study with the aptamer. So this is the aptamer. This is the metabolite. Okay. So what is being studied is the binding energy. Okay. So from there we can deduce, okay, whether the selectivity is good, okay, whether the binding is good. Okay. So how do we also test that? We need to put other than ATCA and then check whether the aptamer will bind to that other than ATCA. So don't worry about that. Okay. There are some ways to study the binding selectivity. Okay. So this is what aptamer can do. Okay. And I also would like to introduce to you this is called molecular imprinted polymer. So this one is considered chemical marker. Okay, just now is ionophore, right? So this is chemical marker. So what is molecular imprinted polymer? Okay, this is an example of what we have done. We want to detect penicillin, okay, in non-penicillin drug. Okay, supposed to be the non-penicillin drug is clear from any traces of penicillin during the production. I don't know, probably there is some cross-contamination probably. So we try to detect penicillin in the non-penicillin drug, okay? So this is the structure of penicillin. And this is the main characteristic, the main functional group of penicillin. We have what we call as beta-lactam, okay? So in order to design this chemical marker, okay, this is how the design process. So it's not that difficult, but we have to do a lot of experiments, okay? So this is the penicillin, okay, that we actually want to target to detect. And then we have to add with monomer. Okay, I hope that you remember the word monomer. You studied in polymer, okay? So the polymerization process happened. Okay, so you can see that where's the polymerization of the penicillin with the monomer, and then we can take it out. The penicillin G here, which has reacted with the monomer, can be taken out. Okay, once it is taken out, this site is empty, of course, but it creates a specific template only for penicillin G, in this case, penicillin G. So it's like a molecule that we design to be specific for penicillin. So how do we use it? Okay, so this is the process. It's not that difficult. Okay, and this is how it looks. It's actually a powder. Okay, I don't have a picture. Okay, right. Yeah, but in morphological studies, actually, it's just the particles. But of course, we cannot see the the cavities, but we design the cavities to be specific binding for penicillin G. So when they interacted specifically, remember the DNA, the antibody. So now when you have specific binding, you can detect the interaction between the imprinted polymer and the analyte. Okay, so this is what we call as chemical marker, molecular imprinted polymer. Okay, again, it has to be studied whether it is selective towards the analyte that we want to study, how good the binding is. So all this requires research to be done. Okay, so I hope that you have more understanding on the production of biomarker and chemical marker. Let's look at nanobodies. Okay, I have introduced to you antibodies. Yeah, you know about antibodies already, but the term nanobodies probably not familiar. 8.7. So what is nanobody? Okay, remember the Y shape of antibody, and this is where the specific interaction with the antigen. Okay, so instead of using the whole structure, because this part does not interact with the antigen, only this part. Okay, so it can be designed. We can synthetically, not to say synthetically, again using recombinant process, we can just produce this one. Okay, apart from using the whole structure of antibody, we can design to have this. Okay, and this is called nanobody. Okay, of course there are advantages. Okay, why people are producing nanobody? Because there are advantages. Okay, one of the issue using the Y-shaped antibody is we have to maintain the orientation of standing up. You know, the Y, sorry, the Y shape has to be like this. If not, then the antigen is difficult to be interacted with the Y shape. If it is lying down or sideways or something cover it up, then you will not get the interaction. But if you have nanobody, okay, it is more, it's not prone to orientation. It's free. It can search for the antigen easily rather than the antigen has to search for this area. So that's why probably nanobody is giving an advantage. Okay. One last one is what we call as CRISPR. So somebody, I think Helmi mentioned about, is a gene editing technology. Yes, it is gene editing technology. Can you excuse me for a while? Yeah, just a moment, Jason. All right, sorry. Okay, so as Helmi said, CRISPR technology is gene editing. So gene is about, yeah, there is translation here. There is subtitle here. Let me remove the subtitle. All right, so the concept of biomarker using CRISPR technology for development of biosensor is similar. It's about the Caesar, the gene editing. Okay, these are extra knowledge, yeah. So this is something that just to introduce you. Okay. Okay, so this is CRISPR, the full CRISPR is clustered regularly interspaced short palindromic repeat. Yeah? Okay, so for treatment nowadays, yeah, we heard about that the CRISPR can edit the gene of cancer gene, can edit the gene of heart disease. Those are possibilities. I read that there are some success story, yeah, to remove genes that leads to some disease, but I'm not so sure. Okay, so how do we use CRISPR in biosensor? This is very complicated even for me. Okay, so the first and foremost, there are three parts. One is the production of this. Yeah? This is called CRISPR RNA Cas13 complex. So I don't want to discuss how we produce the complex. It's very difficult for me, even for me to understand and not to explain to you. All right. So this is probably sample. Okay. And now we come to the microRNA biomarker. Okay. So now we come to the term microRNA biomarker. What's so special about microRNA biomarker is it is abundance. Yeah? It is abundance in sweats, in tears. Okay. So remember that we do not want to have an invasive sampling. Yeah? We don't want to like, especially blood, to draw blood. So by using biomarkers like microRNA in sweats, okay, it makes us easier to sample, to test. You don't have to feel pain because it's sweats. Yeah? So that's what's so special about microRNA. It is abundance in terms of everywhere. All right. So the design of this, okay, you can see the blue color. Okay, it is also matching the microRNA sequence. Okay. So now let's have a situation where there is no target microRNA in the sample. Okay. So the CRISPR system here cannot find the target microRNA. So when it does not find the microRNA of the specific sequence, then the whole process will be like this. Okay, so I don't want to explain this. Okay, we will learn later. Okay, but there's no editing being done. There's no gene cutting, there is no gene editing will be done. But let's look at if the system, the CRISPR complex, find the microRNA of interest in the sample. Okay, you can see the blue color is similar to the microRNA sequence. So once it finds, it will trigger, it will be activated. Okay, the CRISPR RNA will be activated. And you can see some part will be cut. Yeah? This is what we call editing. Okay, so this is actually a scissor cutting the gene. Okay, so of course, when there is gene editing happening, there is different kind of signal generation that can happen. Yeah, if you're interested, can I can give you this slide, yeah, and you can have a detailed reading based on this paper. So it is based on a series of sequence, gene sequence will be edited. Okay, so this is quite new, right? The CRISPR itself is not that new, but the application of CRISPR in development of biosensor is quite new. Okay, right? You can see that it can also be used with rapid test kit. Okay, and this is we have, I mean, myself has organized a talk on CRISPR technology. If you're interested, you can find this title, Webinar on LAMP and CRISPR Technology by NAY Biosciences and Heart. And you will find this talk on the CRISPR technology. Okay, all right. I know it's not that. It's actually very foreign topics to be discussed, but I hope this is something new that can give you extra knowledge, right? Okay. Okay, I think we have covered 3 general biomarkers: DNA, antibody, aptamers, and we also have covered nanobodies, okay, ionophore. We have covered molecular imprinted polymers, and last but not least, we also discussed a bit on CRISPR. Okay, so these are all biomarkers. Okay? These are all bioreceptors, which is the most important. As I mentioned, this is like the heart of the whole sensor. Okay, so the development of these biomarkers or chemical markers itself requires 2 or 3 years. It might be 5 years, depends on your funding, depends on your equipment. Okay, right. So I hope the most important part is you understand all this difference between DNA, at least DNA, body, and aptamer. Okay, I think it's enough. Okay. Starting next week, we are going to do some group activities. I usually do it for 8 to 9 sessions. Probably you will be in a group, in a separate room. Okay, we will discuss some scenarios and you will present a bit. Okay, I hope that you will be actively participating. Yeah, all this while I've been talking. So next week, please be prepared that you also have to contribute to the discussion. Okay, and don't skip a class. All right, any questions before we end?
Prof,
Yeah.
I have a question. So based on the schedule that you sent us a few weeks ago, I noticed there's some face-to-face class in UTM. So is it the same timing?
Okay. Face-to-face class, I put it there, is for the purpose of those who wants to come. Okay, it's not compulsory. It's flexible, meaning that if you wish to discuss in detail, for example, and you wish to understand something, there are certain concepts or there's certain question, whatever question related to this subject, and you want to see me, please do text me first. I open from Monday until Friday, but it is based on we have to text me, we can decide on the timing. So it's not like it's going to be like Friday 6 to 9. No, it's a special face to face. Okay, do you understand? Hello.
Yes, Prof. Yes, Prof.
This is something that
Thank you so much, Doc.
yeah, a special session if you need to, for those, especially those live near, or you actually in UPM, you want to know more. Okay, I can show you devices in our lab. Okay, you can, if you're interested, you can come. Okay, so that's this what face to face in the schedule. is by appointment. Okay, right. Any other questions? All right, if you don't have, okay, right. Okay. I think that's all. So thank you, everybody. Okay, have a nice rest. Bye.
Thank you, Prof.
Thank you, Prof.
Thank you, Prof.